working with Dr. David K. Foster at Messiah University in our Department of Biological Sciences. And today, I'm here to try to make you guys as excited as I am about bluebells. How many of you are familiar with the Virginia bluebell? It's a beautiful, beautiful plant. It is a perennial in the Boraginaceae family. Remember the forget-me-not family. How many of you are familiar with the method of tissue culture, plant tissue culture? By show of hands. Fantastic. Okay. I get to start from scratch, which means it's great. So, back to Virginia bluebells, the plant that I am working on. They're native to eastern North America, including Pennsylvania. They're widely used as exterior home decorations. And classically, we're going to see or tuber propagate them. Why aren't we okay with plastics? Because we're difficult. No, because there's a low success rate of a special white variety that we have on campus at Messiah University. Only about 80% of the time or less, usually much less, does seed propagation actually give us that variety. And so we want to make those numbers higher, and we want to make this a more industrial scale type thing. So our problem is tissue culture. Our lab has tried for the past almost decade to initiate these guys into tissue culture, and we've been unsuccessful, except for one singular case out of thousands. So, tissue culture, the goal is you're going to take them in a lab setting. You're usually going to take a cutting of the plant, put it into an agar-based media with all the necessary nutrients and things, and grow it from scratch, right? This is actually going to do it a lot faster than, say, seed propagation, which is why it's so valuable to us. The overall goal of plant tissue culture is somatic embryogenesis. What are somatic embryos? They're essentially the things that form new plant organs, new shoots of the plant. So your first goal is going to be to de-differentiate the cells and form essentially what looks like a plant tumor. It's very interesting. It's just undifferentiated cells. And those tumors can form roots, and then you're going to form the somatic embryos, which look like little heart shapes. They're real pretty and cute. And then you're going to form new plant organs, which is the shoots. Big picture here, small amounts of cells are used to provide an entire viable plant. In tissue culture, your media is key. There are two key hormones to tissue culture, cytokines and auxins. Cytokines produce shoot growth, auxins produce root growth. We want to find that specific ratio, what makes a good shoot versus root dynamic here. Classically, you're going to have browning that is going to indicate usually negative activity. We don't love browning. It sort of gets in the way, so that is something we're going to study here and see what is that perfect balance between browning and callus tissue formation, which is the first step of creating a tissue culture. And we are using algae-elated tissue because, quite frankly, we ran out of options. When it comes to non-elated tissue, it doesn't work for us. And so we grow this tissue in the dark, hoping that there is some kind of light switch or some kind of cold-tolerant switch that is working with us. When flowering initiates, the tissue is no longer viable for culture. This indicates that we have to use a specific age range, usually 1 to 2 years, but we don't know that for sure. This is just an observation we made, so that's another thing that I am confirming. And we want to figure out what that pathway is for flowering that causes the rooting and shooting pathways to cease. It could be a genetic moment, we don't know. 3 hormones are used. TDZ is a cytokinin, 2,4-D is an auxin, IBA is an auxin. So, TDZ, shoots. Everything else, roots. Our methods, again, talking about the age difference, we're using 3 different age categories: 0 to 1 years, young plants; 1 to 2 years, middle-aged plants; and 2+ years, old plants. Those old plants, we call them old because they've already got preformed flower buds. And remember, that flowering switch is something that we are concerned about. We don't think it's going to allow us to initiate into tissue culture. We're using 3 different combinations here, and I'll talk about those as I get to the results for those sections. First result section is our IBA section. These are using either 1 ppm or 2.6 parts per million of IBA only. We got tons of callus tissue. This was so exciting for us. I remember the day my professor walks up to me and he says, "Ty, you should check on your plants. There's some fun activity going on." So I run down there and I look. I got callus tissue. I got plant tumors. I'm like ridiculously excited about this. I know, it's really fun. And so the other thing is the browning was super delayed. It didn't start browning until almost a month after we put them into culture, which is really good because it doesn't interfere. And here are just a bunch of various pictures. You can see roots, you can see callus tissue, which is these blob-looking things there. They're great. In terms of the 1 ppm 2,4-D with 1 ppm TBC, the literature suggested that in other species, if we dip them in the 2,4-D, and then put them into a TBC media, they proliferate, you're gonna get the rooting, and then you're gonna get the shooting, and it all works out really nicely. Didn't work for us. Bluebells don't do that for some reason. Bluebells are sneaky little things. The thing that I spent the most time on was 1 ppm IBA with 1 ppm TBC. I know it's a lot of numbers, but let me try to break it down. Each group here, orange is the young group, middle is the gray group. If you remember, middle is the one we had the most hope for, and yellow is the older group. In terms of browning, it's super variable, and we do have significant browning across all the different groups. It depends on the degree of it. There wasn't really any statistic difference there. But when it comes to callus tissue, what do you see? Does everyone see that gray bar in the higher categories here? That's important. We're getting the higher callus tissue formations in those middle plants, thus at least giving credence to our hypothesis, right? Really good. So preliminary determinations here. First off, evolution wasn't that magic switch we hoped it would be. It doesn't mean it's not a factor. It does mean it's not the factor, the deciding factor. It doesn't really work best to get the roots first, which was what we've been doing. We've been trying to take a viable piece of plant tissue, root it, and then let it survive. That's how we've had one success thus far. It was the only success we had, and we haven't been able to repeat it. So we are just about ready to give up on that perspective here. Final thing that we recognize is this idea of transitional competency. If you look at the plants, the 3 age groups, some are too young, some are too old, and some are just right. This transitional competency puts them in that transitional period between forming the preformed flower buds and being juvenile plants. We think that's where they're most competent for callus tissue formation, and thus we think those are our best chance at using viable tissue. In discussion here, what do we want? What's our success rate, right? It's roots and shoots. We want one plant to survive that has both roots and shoots from something that we have done in tissue culture. This is great. This is really great in theory. We've done it once. Again, out of thousands of trials, it's happened once, and we still haven't figured out this system. Next, we're searching for somatic embryos. Those little heart-shaped things that I told you earlier, where they form the shoots, you can only find them under a microscope. I've got something like 90 samples in my lab right now with callus tissue. I'm going to have to go through each of them and try to look at them under a microscope and find a very tiny little heart-shaped thing on this glob of callus tissue. But if we can find that, we have hope. Because if we have hope, we have somatic embryos, we get shoots, we get those new plant organ developments. When I say reverse order, we're going to try to get the shoots first and then the roots, seeing as we're sort of moving past this idea that we can take an already shooted thing and put roots on it. Another thing we want to try is the evolution variable on IVA. We've shown that IVA forms really good callus tissue, like really good callus tissue. So we're going to look and see if we can get some callus tissue on non-evoluted tissue, on just wild tissue that has been grown up in the sun. And then finally, we're going to do a little plant surgery. We're going to take some of the callus tissue that are in these giant globs. We're going to cut it off, put it in TBC only media, and hopefully form roots and encourage those somatic embryos, even if we can't see them, to produce shoots. It's sort of a foolproof method if it works. I'd like to acknowledge Dr. Jamie Foster for his mentorship and his work with me this semester. I'd like to thank Masai University's Department of Biological Sciences for their funding and for the space that I've used. And I'd like to thank Gannon University for hosting us. So, any questions, guys?
So, for the one successful plant, did you end up like transplanting that to soil? Is it grown?
It did transplant to soil. It is no longer a viable plant as of now, but this was something like 4 years ago. Long while ago. Yes?
Yeah, how specific is this research to the movement of bluebell, and what are the implications for other plants?
Sure. So this is hopefully going to open up a bunch of other plants within the genus and related genuses. One of those plants is trilliums. I don't know if any of you are botanists or anything like that, trilliums are expensive. And if we can figure out how to get trilliums into tissue culture, that's a win for us. I mean, that is a big economic deal not only, but it's also an industrial thing.
So yeah, so this isn't really like this is mostly industrial and economic and like for cultivation in terms of gardens and things.
Sure, yes. And biodiversity, as your project was as well, yeah.
Have you tried any liquid culture with the somatic embryogenesis? Because I know they do that for carrots, and they grow it on a shaken suspension. And have you looked into any of that?
We're looking into it actually right now. So that is one we are seriously considering. The problem with that is that you don't get that solid suspension, and so it can mess with the fine root hairs. They don't like being touched, which is a problem that I have discovered accidentally when messing with my samples. Yeah.
So you talked about how bluebells are used for like landscaping, things like that. Do you know if there's any medicinal properties of it that would be important?
Not super well studied, so potentially yes, but we don't know. Yeah.
So when you're like inducing a callus, are you using for all your samples the same parts of the plant, or are you going in different parts?
So I'm taking a pair of scissors on emerging plants. I'm cutting and snipping, so it's all either stem or leaf tissue, but all of them have stem tissue in them. And the other thing is, it's all in the same. The other thing that we keep the same is the media. It's Mershagi and Scrooge media with all the needed nutrients for the plant. So the plant's not missing anything. It's just that added hormone. Yeah.
Are the somatic embryos only going to be on the outside of that cell mass?
Of the callus tissue?
Yeah.
Yes, sir.
Okay, so it's like they won't be hidden inside.
If they were hidden inside, they're going to get choked out.
Right, okay.
Any other questions? I love this. Yeah.
Before you prepare like a viable part of the thing, can you examine them under microscope? I'm just curious.
Examine them for much, right?
When you take it, do you examine it under a microscope or anything to see?
Not under a microscope, but you actually introduce something very interesting. And our biggest problem is contamination. The plant has 2 cell walls. And so classically, we will get something called hyphae bacteria, which are my least favorite thing on this planet, worse than aphids in my opinion. Because when you snip that cell wall, and if the hyphae is in between the 2 cell walls, what do you do if not release the hyphae? And then the hyphae infects half my samples and my data size goes down significantly. It's not fun. Yeah?
Did you do any other plant tissue culture before you started working with the bluebells?
I like this question. I'm a pre-med student, so no. But my professor managed to, not managed to, I fell in love with it. I've always kept a garden myself, and so doing this kind of thing on a really scientific level, combining my love for gardening with my love for science, it was so exciting.
What species did you practice on to get your sterile technique down?
Where's my Venus flytrap person? Is she still in here? Because it was
He's in the other room.
Venus flytraps. Those are also buggers, but I love them. Yes.
So which is harder, cultivating these tissue cultures or tumor cells?
Tumor cells are easier by far.
Right.
So far. He's my cancer problem. Any other questions?
Time for one more.
Yeah.
Got any tips? I'm doing this next semester.
Are you really?
Not with bluebells.
Okay. Sterile technique. If you didn't, I spent an entire semester on solely sterile technique and like the Venus flytraps. If it's not an issue with your cell wall, you probably shouldn't have contamination. So like why I'm wearing all that random stuff on.
Tyler, do you add antifungals?
No.
Why not?
We tend
Your
to
major contaminant?
yeah, it tends to interrupt is the problem. Interrupt with the cell cycle of the plant. When you're coming to rooting and shooting with the auxins and cytokines, they don't like each other.
They're too closely related metabolically.
Right, they don't like each other.
All right, thank you, Tyler.
Thank you.